Hello, sir. Hello, guys. Good afternoon. This is Dr. Pooja, and I will be taking this session for you guys on cell as the fundamental unit of life. All right. Let me see if we are all connected. Yes,
Hello,
we
sir.
are.
Hello, guys.
Yeah, I'm sorry. So, guys, today I see there are a few people already. Twinkle, Akula Prasad, Tanima Pati. So, guys, welcome to this session. And in all my sessions, I try to, explain things to you in more diagrammatical manner so that you guys can understand everything properly and clearly. And I hope I'm audible to you all. You can see me, you can listen to me, right? If not, then let me know. And also, if you have any questions, any queries, please feel free to type it in and I'll be more than happy to answer all your questions. Okay, so today we'll discuss cell as the fundamental unit of life. And guys, a little bit about myself before we get into the details of this chapter. Okay, you need to know your educator before you learn something from your educator. Okay, so my name is let me take a quick color. So my name is Dr. Pooja, and I am a former scientist and lab director from United States of America. I was there for almost 8 years, and I've done PhD in biotechnology. So I have a very good knowledge of any stream of science, be it physics, chemistry, or biology. So if you would like to learn more from me, then you can go ahead and sign up on Unacademy's platform, because now I am a science educator on that platform. Otherwise, I'm also online fitness coach. I take care of your health and fitness. And also, apart from this, on our Unacademy's platform, we take care of not only the academics, not only the science chapter or the studies. We also take care of your health, your fitness, your mental health, and everything. And I have all these special lectures lined up on Unacademy's platform. If you want, you can go ahead and sign up there for the free lectures that I have. So here is the link to the same, and you can go ahead and, as I said, sign up for the same. All right, so now another good news for you is not only 9th, 10th, 11th, 12th, we have now 6th, 7th, and 8th also available. You can subscribe for these classes as well. And so what is the beauty of Unacademy is we have live classes for all the subjects daily. We have weekly quizzes, we have doubt solving live sessions, we have practice question sessions and test series. So and if you want to go ahead and sign up for Unacademy's platform, you can use my code, which is PS13, for extra 10% discount. Correct, guys? I will be talking more about like how you can sign up, because we are both in Hindi and in English language. So whatever you want, you can just sign up and we can help you with the same. And here are the different subscription plans. You can go for 1 month, 6 months, 12, 24, or 36 months, totally up to you. And also 48 months, right? So as we are having classes from 6th through 12th, so you have a plan for like 6 years. So it is up to you how you want to do it. And again, on top of all these numbers that we have here, if you use this code, you're gonna get extra 10%. Okay, so you can go ahead and try for that. So guys, this was a quick overview about like how we work on Unacademy's system, and how many different options you guys have to avail from, right? So now we will talk about today's topic, which is going to be super interesting for you. Okay, so let's get started. And let's put your thinking caps on and let's crack it. Okay, so whosoever is there in the chat with me, you guys can always send me your messages, send me your text, and I will report on that so that I can answer all your queries, all your questions. Okay, so guys, let's get started on the topic cell, and I will definitely make everything super clear to you. Okay, so first of all, what is cell? Okay, so that is the basic question that comes to everybody's mind. What is cell? Right, so guys, when we talk about, let's say, when we talk about any kind of let me take another pen real quick. So when we talk about, let's say, chemistry, okay, and in chemistry we talk about elements, and when we talk about elements we go back to, I will not get into the discussion of like the particles and all. I will directly say we came down to the term atom, right? Like whatever we see around us is made up of atoms, and atoms, they further have subatomic particles like electrons, protons, and neutrons, and that is like something, some topic that I have covered in my chemistry lectures. But here, if atom is the basic unit of, let's say, in chemistry, we go down to atom and talk more about the elements in details. How about the basic unit of living organisms, right? How about like what is the basic unit? What is the fundamental unit when we talk about, living organisms? So now we know, now it's easy for us to say, okay, it is cell. But have you ever thought about it, like when these scientists, they were in a very original state, and there were not many equipments available, there were not many microscopes available. How did they do all this research? So there was a scientist named Robert Hooke in 1665. He just observed cork cells or cork under his, very basic microscope. Okay, and to his surprise, he saw that there were honeycomb-like structures, right? And he thought, what are these compartments? So the compartments means the little room. Cell means a little room in Latin, if you talk about. Okay, so from there he got an idea, like whatever we see around us is made up of some kind of little compartments. And later on, when they discovered, advanced microscopes, electronic microscopes, and all other things, then they got to know more about cell, and that is what we will discuss in more details today. Okay, so there is, if you get this question like who discovered cell, and in when did he discover, then here is the answer to that. So hold on. So this is like, even if you get a chance to peel the onion skin, or you get a chance to get skin out of your cheeks, okay, and you get a chance to strain it with some dye, okay, for example, maybe iodine solution or methylene blue, okay, so what will you observe under the microscope? If you observe, this is what you're gonna see, right? There will be different compartments and these little dots over here, right? And I will discuss in a minute like what are these little dots and what are these called. Okay, so this was all about like the how the cell was discovered. So as atoms are the basic unit of, I would say, all the elements, molecules, compounds, okay? Similarly, if we talk about the basic unit of life, we call it cell. And guys, don't get confused here, because every living thing, if we say every living thing is made up of, let's say, cells, and these cells are again have carbs, proteins, fats, etc. And these are again biomolecules, okay? So these biomolecules are, if we keep on dividing, yes, absolutely right, Rajmani. So ultimately we will get down to elements and further down to atoms, right? But we are not going to that much of detail, because we don't want to know whether we are made up of carbon atoms or hydrogen atoms and all those things. We just need to know, like, our bodies are made up of, you go, cells, right? And we will stop here only, and we will learn more about, like, if we are all made up of cells, what are the different parts of the cells, what are the different functions of the cells, okay? And that is what I'm going to discuss in more details, because many people, many children, they get, confused with all these different components of the cell, okay? So here you go. And before we get into the details, there are a few more scientists that you will have to remember, and their important discoveries as well. So Robert Hooke, as I said, he discovered the cell, and the nucleus was discovered by Robert Brown. Then Mr. Leeuwenhoek, he said he found, like, in pond water, he found certain free living cells, okay? Then these cells, they have some kind of clear jelly like substance, and Purkinje, he called it protoplasm. So guys, these are like for your 1 mark MCQs, okay? So these questions could be there in your examination, like who discovered the cell, who discovered the nucleus, and who found the free living cells in pond water, who coined the term protoplasm, who gave the cell theory. We have Schleiden and Schwann. Then we have like cells, they occur from the pre-existing cells, and that was given by Virchow. So these are a couple of scientist names that you will have to remember, right, if you want to score some marks in your MCQs, okay? So it was just a quick overview. Hello Shivani, how are you? So guys, I left these couple of sheets blank for you only, so that we can discuss, but let's go over these one by one, okay? So now, as we know the very basic idea of the cell, like what is there and who discovered the cell, and yes, Tanima, you are absolutely right, okay? He said omnicellula is cellula, which means the cell, they are they don't like get originated from nowhere, they get originated from the pre-existing cells, right? So that is what Virchow gave, okay? He gave the refined theory. So now guys, we will discuss about different components of the cell, and these are, like, we know that cell membrane or plasma membrane, nucleus, and cytoplasm. Okay, so quickly what I will do here is I will explain. There is an analogy that you can do. That is how I explain it to my plus students, so that you guys have an idea like how the cell has this compartmentization and how different organelles are present. It's just for a quick comparison. Let's say this is your house. Okay, you have a door, right? You have, this is your door, and you have walls, you have, let's say, windows, right? Quickly I will just explain. Then you have different rooms where you perform, different functions. Let's say you have kitchen here with you have some kind of ACs, TV, right? All this is the empty space in your house, right? Then you have your parents, your mom and dad, right? So this is what we will do now. Compare it with the cell. Okay, I've drawn a cell quickly. So as in our house we have walls, right? We have these walls. Similarly, in case of cell, we have plasma membrane. Okay, that is the protective covering that we have covering our cell, right? And this, as the walls of our houses, they have doors and windows to allow only few selective things inside, right? It is not like anything can get into your house, right? Only few selective things can get into your house through your door and wall and your windows, right? Similarly, plasma membrane is also selectively permeable. Selectively permeable. It will not allow each and everything to get in and out of the cell. Okay, it has control on that. Why? As we have doors and windows in our house, similarly in our let's say, let me take another column. Similarly, for our plasma membrane, when we talk about the plasma membrane details, then I will explain. So it is a bilipid membrane, and it has protein channels. I will explain in my next slide maybe. So these channels, they allow only selective items to get in and out of the cell. So that is why our cell is, or our plasma membrane is, selectively permeable. Okay, so that is one thing. And then, hi Asmita, then secondly, just like we have all these different this empty space in the house, similarly we have this space in our cell. Okay, so this space, we call it cytoplasm, right? So yeah Tanima, I will explain like what all is there in the plasma membrane. Okay, so we have, just like house we have empty space, just like that the cell has this jelly like substance. And in this jelly like substance, like in our house we have ACs and TVs and all other gadgets. Similarly, our cell has all these gadgets, and let me draw a couple. For example, we have mitochondria, right? Another mitochondria, right? Similarly, like we have equipment in a house, our cell is also equipped with all essential things, right? I will explain, Tanima, give me a second. Then we have endoplasmic reticulum is present in the cell. Okay, so these are like all different items present in the cell. So your endoplasmic reticulum, I will draw much neatly in my next slide maybe. So they have ribosomes on them, so that is rough endoplasmic reticulum. When they don't have ribosomes on them, that is smooth endoplasmic reticulum, right? Then we have Golgi-Paris, right? So these are the different parts of the cell, just like we have it in our house. Then, as I said, we have mom and dad, like for our control. Similarly, in the cell, we have this, what we call nucleus, right? So this is very much similar to our house, how we have different things in our house, we have different control units in our house, we have different equipments in our house. Similarly, our cell has everything which is required to sustain independently. For example, mitochondria, they are the energy house of the cell. Endoplasmic reticulum, they make important proteins and lipids. Okay, your Golgi bodies, they pack all these proteins and lipids and they send them to their places. Then we have nucleus, and that is there for the control, right? So because now everything is been there in the cell, we have protein, lipid, packaging, we have lysosomes as well, right? We have ribosomes as well. So all these items present in the cell, which means cell can sustain independently, right? If cell can sustain independently, that means we can say cell is the basic or fundamental unit of life. Correct, guys? This is the reason we call cell as the fundamental unit of life, because it can exist independently. Okay, it can live independently. It has all the essential items present to live independently. So let's start with, so that means we'll have to cover three major points. One is plasma membrane. Second, we will cover nucleus. Third, we will cover organelles. Right? Rajmani, I'll explain in a minute, bacha. So these are the three main important topics that I will be covering today. Okay, so first of all, my first slide is plasma membrane. Okay, so this is a very magnified picture of how plasma membrane it looks like. So guys, when you have a look at it, let me explain it in next slide and I will get back to this. So plasma membrane, if you look at it, is made up of lipids. So here we go, right? And these are hydrophobic, hydrophilic, you all know that. So this head is hydrophilic and these tails are hydrophobic, right? So this is the lipid bilayer model of plasma membrane. And not only this, plasma membrane has many other items. For example, if you see closely, it has these protein molecules or protein channels are there. So these are the ones which help in, selective movement of certain things across the plasma membrane. For example, this is something extracellular, which we call outside the cell, and this is cytoplasm on this side, right? And let's say if I draw this cell, it will be something like this. I have, let's say, magnified this part of the plasma membrane. All right, so if I show you now, this is what it looks like. All right, this is the very basic plasma membrane picture that I've drawn for you. And if we go little back over here, so here are more details, right? So this is the phospholipid bilayer, okay, with all these red color are the heads. This is one phospholipid molecule. It has this hydrophilic head, and the tails are hydrophobic, correct? So so you see whole plasma membrane is made up of this. And I have already covered this: hydrophobic tail, hydrophilic head. There are many carbohydrates present. There are different kind of proteins are present. There are surface proteins present. There are globular proteins present. Okay, these are kind of pathways. There are peripheral proteins. Somebody was asking, ma'am, cholesterol is present. Yes, cholesterol is also there. So this is the detailed structure of your plasma membrane, right? So this is how it looks like if you really magnify your plasma membrane. And it is a phospholipid bilayer, and it has, you know, proteins, integral proteins for to give it a property of, you know, selectively permeability. Okay, so this was about plasma membrane, guys. And now let's move on to nucleus. Okay, so nucleus is the controlling unit. It is the, okay, Amit, welcome to my class. So nucleus, Amit, please stop it, is the controlling unit of the cell. Okay, it controls everything. Why so? Because nucleus, it has genetic material, right? For example, in our case, it is DNA, deoxyribonucleic acid. Okay, so this DNA is the genetic material. It carries information from parents to their offsprings. And this DNA over here, it helps in different kind of protein synthesis. Okay, right? So we'll talk about how this protein synthesis takes place. When DNA makes another copy of DNA, that is called replication. Not here, this is not there. Then we have transcription, because this protein formation needs some kind of mRNAs, rRNAs, tRNAs. So these are all made by DNA with a process called transcription. And then we have a process called translation, which is the protein formation property of, you know, from DNA. I mean, it is a very complex system. I cannot cover all these points here, but still you need to understand how DNA is the controlling unit, because it is the one which helps in formation of different kind of proteins, and through the transcription and translation processes, not replication, because replication is the process when cell division takes place. Okay, so here when scientists, they further started coloring the cells, they started seeing all these, you know, dotted structures in the center, and these were the nuclei. Okay, from there they got an idea, okay, like, because nucleus is present, so the these dot-like structures are there, they call them nuclei later on. And Robert Brown was there. Yeah, chromosome is the like condensed form of DNA when the cell is ready to divide, and it gets wrapped around, you know, some kind of special proteins, and that is called condensed form of DNA, I would say, called chromosomes. Okay, so here this central unit over here is nucleus, and, you know, over here this yellow thing. Okay, and then I will explain why it is surrounded by this purple kind of thing. This is your endoplasmic reticulum. Okay, we'll talk about it in a minute. So now if we have covered plasma membrane, we have covered nucleus, right? Yes, those are histones, Divya. Now we will talk about, there are different kind of H1, H2, H3. Those are the names of the histones. And the third part is now we will talk about cytoplasm, and basically in cytoplasm we will talk about, you know, your organelles. And the important organelles that we will discuss is your endoplasmic reticulum, Golgi bodies, then we will have mitochondria, then we will talk a little bit about lysosomes, right? Then we have vacuoles, right? Then yeah, cell wall we will just touch base a little bit when we talk about, you know, plant cell and animal cell differences. Okay, so these are the hello Perth. So these are the points then that we will discuss when we move little further. So guys, now you will have to understand one thing here, as we have yeah, not that much about centrosomes and centrioles and all those things, because at this level when you are in 9th grade, you really don't have to focus on like what are the centrosomes and centrioles and nucleolus and all those things. Okay, in your higher classes, of course, you have to learn more about all these different organelles, but at this class, at this level, I guess this is all sufficient. And I can, I mean, if you want to get into the details of those organelles, we can talk into more details. Okay, so guys, we'll see the endoplasmic reticulum. So now we have all these, you know, 10th class I take every day 8:45 to 9:45. Guys, I'm a plus teacher plus educator on Unacademy's platform. Okay, I teach you guys physics, chemistry and biology, 9th and 10th both. Okay, every evening 7:30 is my 9th class, every evening 8:45 is my 10th class. Okay, so you guys are more than welcome to come and be part of my, you know, live sessions, because my live sessions are just awesome. Okay, these YouTube sessions are little different from the live session, so I would strongly encourage you guys to come on Unacademy's platform, and then you can learn, you know, physics, chemistry, biology with me on that platform. Okay, so guys, as we are moving on to the organelles part of our cell here, no Divya, this is not about the higher concepts, I'm sorry. This is all about very basic concepts. Okay, this is for not for your NTSE. Okay, so endoplasmic reticulum is present closer to the nucleus. Can you tell me, Divya, why is it present closer to the nucleus? Do you have any idea? And endoplasmic reticulum, when we discuss it in more details, it has, some of it has ribosomes present and some of it does not have ribosomes. The one with ribosomes, hello Bhumi, happy birthday to you, with ribosomes is rough endoplasmic reticulum. No ribosomes is smooth endoplasmic reticulum, and I will discuss about these in a minute. Okay, then we have Golgi bodies. Endoplasmic reticulum is basically, I would say, the production house of the cell. It helps in formation of proteins, different kind of proteins, and then lipids. So I would say endoplasmic reticulum is the factory inside the cell. Okay, just like you have kitchen in your house, similarly endoplasmic reticulum is kind of kitchen for your cell. Then Golgi bodies. So Golgi bodies, when we are done with making the proteins and lipids, there is no point if these lipids and proteins are sitting inside the cell. They have to reach out to the place where they can function properly, and for that Golgi bodies, they act as the transportation system. Okay, they help as transportation system. What do? They make certain changes to the proteins and lipids. They process them and they send them outside the cell if it is an extracellular protein, or within the cell if it has to be used by the cell itself. Then mitochondria is the energy house of the cell, because it helps in production of ATP, which is adenosine triphosphate. Then your lysosomes, they have very strong digestive enzymes, and we call them, if anything from outside gets into the cell, so it reacts with all these digestive enzymes and it kills it. But let's say sometimes when cell is not properly functioning, then lysosomes, they eat up their own cells, right? Then that is why it is called suicidal bags, guys. Okay, so that is why we call lysosomes as the suicidal bags. Then vacuoles, they give turgidity to the cell, and there are big vacuoles present in plant cells, and there are small vacuoles or very, very minuscule vacuoles present in animal cells. Okay, basically they are the property of the plant cells, right, guys? So quickly I just summarize what are the different parts of different organelles present in the cell, and now let's discuss them one by one quickly. Okay, so here is your endoplasmic reticulum that I'm gonna discuss. Let me take some other color. All right, I'll take red. So guys, here is your nucleus, and if you see, your endoplasmic reticulum is surrounding the nucleus. Okay, that too your rough endoplasmic reticulum. Why rough? Because it is having ribosomes on it. So guys, there should be a question asked by you, why rough endoplasmic reticulum is present right next to the nucleus? Because what happens is your DNA is present in the nucleus, right? Your DNA is there in the nucleus. Let me write it down somewhere. Okay, so DNA is present in the nucleus, and your DNA, it helps in protein synthesis. How? Okay, so how it helps in protein synthesis? Because yes, Mukul, absolutely right. So DNA, it gives rise to mRNA, messenger RNA, right? And this messenger RNA comes out of the nucleus, and with the help of ribosomes and amino acids and tRNA and other biochemical compounds, it forms this mRNA. It helps in formation of proteins. So that means if mRNA is coming out of the nucleus, ribosomal part should be sitting next to it. That is why our rough endoplasmic reticulum. Rough endoplasmic reticulum means these are the plates like structures, but they have ribosomes on them, just like this. So as they have ribosomes on them, this gives them a rough appearance. That is why we call them rough endoplasmic reticulum. Okay, and after the rough endoplasmic reticulum, if you move a little bit further down, this brown structure you see, is the smooth endoplasmic reticulum. Why it is smooth? Because it has no ribosomes, right? That is why we call it. Mukut, my next lecture, as I said, I'm Unacademy educator. So you can find me on Unacademy's platform, and I take ninth grade lectures at 7:30, and I take 10th grade lectures at 8:45 every day. Okay, and for that, if you want to be part of my plus, as I teach you guys physics, chemistry and biology, everything, so you can take the plus subscription, and here is the code that you can use for extra 10%. And trust me guys, all the educators on Unacademy's platform, they are very well educated, and they teach great with all the test series, with all the problem solving sessions, so it is worth joining, right? So I already explained about endoplasmic reticulum, but quickly here. So let's say this is your nucleus, and here is the endoplasmic reticulum present just outside the nucleus, right? So what is happening over here is your mRNA is coming out of the nucleus, right? Let's say this is the mRNA, right? It is coming out of the nucleus, and then you have ribosomes present, along with the ribosomes and along with the other components, as I just discussed, it will start making proteins. So proteins are just the chains of amino acids, guys, right? So these are the proteins which are formed, right? Then we have this over here farther down, we have smooth endoplasmic reticulum. I just showed you in that picture. I'll quickly show you one more time. So this one is smooth endoplasmic reticulum further down. Okay, so this is how protein synthesis takes place when it comes to endoplasmic reticulum. And your next, this is like even better picture to explain the same thing. Let me take another color. So here is the nucleus, here is the rough endoplasmic reticulum with all the ribosomes on it, right? Do you see these dots? These dots are the ribosomes present, and this over here is your smooth endoplasmic reticulum. Okay, this over here is your smooth endoplasmic reticulum. All right, so this was quickly about endoplasmic reticulum. Now let's touch base a little bit with your Golgi apparatus. Okay, so this is the nucleus, this is the endoplasmic reticulum, this is smooth endoplasmic reticulum. Nucleus sends messenger RNA to your endoplasmic reticulum. It starts making protein. What will happen to that protein? Okay, if it has been just made inside the cell and it is not reaching out to the parts of the cells or parts of the body, what is the point? Then who processes this protein? Who changes or packages this protein? Golgi body. Okay, so we have these tubules and these vesicles together, it's called Golgi body. Okay, so this is the system which is kind of transportation system, packaging and transportation system of the cell, or they call it traffic police of the cell. They have just named them differently. Okay, so what are its functions? Basically, as I said, Golgi bodies, it helps in packaging the protein. Okay, so let's see in this picture. I have this picture to explain things even in a better way. Okay, But before I do that, let me draw quickly for you. So let's say this is our cell. Here is our nucleus. Right next to the nucleus was our endoplasmic reticulum. Excuse me guys, my drawing's little weird. And then we have Golgi bodies over here somewhere. Okay, so what's happening? Try to understand this picture. Your DNA is produced. I'm sorry, this is your rough endoplasmic reticulum with all the ribosomes on them. I'm really sorry, I just forgot to draw. All right, so your nucleus, this is the nucleus, endoplasmic reticulum, Golgi bodies. Okay, so what is happening? Your protein is being formed in endoplasmic reticulum, and in small vesicles this protein goes to Golgi bodies, and this is the site where it gets modified and processed. Right, now this protein is here present in small vesicles. Now there are two options. Either they go out if these proteins are extracellular, right? This protein will be released out. It can be used by other cells of the body, or this protein can go to other parts of the cell. Let's say it is required somewhere here, right? So this functionality is controlled by Golgi bodies, packaging and sending the protein either outside the cell or inside the cell to some other parts of the cell, right? So let's see it in a picture now. So here is the picture that I just drawn in my previous slide. Nucleus making mRNAs. Those mRNAs along with different other components on basic ribosomes and on endoplasmic reticulum. You see these little dots like structures formed? These are the proteins, right? And these are packaged in the vesicles and they move on to the Golgi apparatus. Now Golgi apparatus is, there are series of modifications going on, and these protein can either go to the different parts of the cell or they can go out of the cell to be used by some other cell of the body. Okay, so this was quickly about Golgi apparatus. Okay, so now is the mitochondria. Okay, so we know mitochondria is the energy house of the cell. We call mitochondria energy house of the cell, and you see mitochondria are present all over the place. You see, right? So we have nucleus, endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi bodies, and here is our mitochondria. And if you see closely, mitochondria, they are, let me draw. So mitochondria powerhouse of the cell. Okay, hello Purushottam Tiwari, for ATP formation. And if we see mitochondria, see, these are double membrane organelles, right? They have double membranes, and these structures over are called cristae, right? So if you and the strange point about mitochondria is they have their own DNA, right? So from here many scientists say, like mitochondria, they have their own DNA, so maybe they are having some kind of symbiotic relationship with the cell, right? Because they have their own DNA, so that means they are kind of independent, independently occurring structures, independently occurring bodies, right? So before we get into the more details, thank you so much, Pratyasha. Thank you. And if you want to learn more from me, then you can go ahead and sign up on Unacademy's platform. You can sign up for the batch courses. Okay, so new batches are starting soon with all different subjects, with all different great timings and test series and crash courses. So you can be part of that. And if you want to learn from me, and you can search, I don't know how many of you are new to my session, you can search for Dr. Pooja, and here is my code that you can use if you want to avail a 10% discount. And I would strongly encourage you guys to go ahead and sign up on Unacademy's platform. Okay, so here as I draw the structure, cristae, it has inner membrane, it has outer membrane. This is the structure of mitochondria. Okay, so it is why we call mitochondria as the energy house of the cell, why we call it the energy currency. Let me explain real quick. So let me rub it off. Okay, give me a second, guys. Hold on. Okay, all right. So what happens is we take energy from the food, and food gets converted into glucose. It burns with oxygen, in presence of oxygen, gets converted into pyruvate. Okay, and this happens in the cytoplasm of the cell. Okay, so now this pyruvate goes to the mitochondria, where pyruvate in presence of oxygen gets converted into, hello Ashu, thank you, carbon dioxide plus water plus energy. So where is this energy getting used? So this energy is used to convert ADPs to ATPs, guys, right? So this over here, this molecule is adenosine triphosphate. So this is the energy currency of the cell. Adenosine triphosphate is the energy currency. Here is adenosine diphosphate. So which means, let me explain it to you. So we have ATP. So this is adenosine. Tri means it has 3 phosphate groups, 1, 2, and 3. Let's say we are performing some kind of function. So what will happen? We'll break this bond, right? When this bond is broken down, it gets converted into ADP. So what is ADP? Adenosine diphosphate, 1 phosphate and 2 phosphate, right? And the energy which is produced here, we use this energy to do our functions. But in return, we are producing ADPs. So that means we need to convert this ADP further to ATP, right? Only then we'll be able to use this energy again, and we convert it to ADP again. How is this happening? This is what is happening. This is what is happening at the mitochondrial level. Mitochondrial level. Okay, so that is why we call pyruvate is yes, absolutely, Harsh. Pyruvate is a 3 carbon compounds, various cell organelles. Absolutely right, Rajmani. Okay, right. So this is all about all the important organelles that we wanted to discuss. Now let's quickly discuss what are the differences between prokaryotes and eukaryote cells, right? We have prokaryote, pro means primitive, original. Eukaryote means the true ones. So guys, if we talk about, thank you Bhumi. So if we talk about prokaryotes and eukaryotes, this is the differences that you will have to explain in some of your examinations, in some of the questions, like what are the differences between prokaryotic and eukaryotic cells. So first difference is the size. Prokaryotic cells, they are very small in size. They are approximately 1 to 10 micrometers, and these are 5 to 100 micrometer in size. So that is the first difference. Second biggest difference is in prokaryotes, the DNA or whatever the genetic material is not surrounded by a wall. It is just present as the entangled mass of the genetic material. It means it is not in the form of nucleus. Nucleus is not there. We call it nucleoid. And guys, strange point is if you start opening this DNA, it will be one circular form of DNA in prokaryotes. But in case of eukaryotes, we have a well-defined nucleus with the nuclear membrane, and the DNA may not be present in one circular unit. It may be present in many fragments, right? And we call them chromosomes. When they condense, and they condense around some kind of protein, so that is what we call chromosome. So that is the second biggest difference, right? Third biggest difference in prokaryotes and eukaryotes is in prokaryotes we don't have membrane bound organelles. They are absent in prokaryotes, while in eukaryotes the membrane bound organelles, whatever we have discussed so far, like Golgi apparatus is there, you have mitochondria is present, smooth endoplasmic reticulum is there, rough endoplasmic reticulum is there. So all these membrane bound organelles are present in your eukaryotic cells, right? And the last difference I would say, in prokaryotic cells you have only one kind of organelles, and that those are ribosomes, right? If you get a question like what kind of organelles are present in in your prokaryotes, then you have to say only ribosomes are present, right? So yeah, size is already there. I've discussed size is there, genetic material is there, it's in the form of circular, and only one I would say strand of genetic material is present, right? So these are the different points. And what about the difference between animal cell and a plant cell? Both are eukaryotes, guys, right? Both are eukaryotes. So that means they all going to have nucleus. They both going to have, organelles. So what are the differences then? Right? What are the differences then? Difference is, number one point is they have plant cells, they have cell wall. Second, plant cells, they have large, you see this vacuole. Guys, I'm always confused between the spellings for vacuoles. Okay, so we have this large vacuole present in plant cell only. Animal cells, they don't either have vacuoles or they have very, very, very small vacuoles. All right. So, and apart from that, because plant cells, they are having this cell wall, which is kind of with the deposition of cellulose, because it has to give strength to the plant. Okay. No such wall is present in your animal cells, right? So this was a yes, absolutely right, Rajmani. They have plastids with chlorophyll, chloroplast. Why? Because they have to undergo photosynthesis. Here we don't have them, right? Because if we have plastids and chloroplasts in us, will be photosynthesizing, right? And that is not possible. We are heterotrophs, we are not autotrophs. So guys, this was quickly all about your what are the different organelles present in the cell, what is the structure of the cell, why we call cell as the fundamental unit. So this was all about cell quickly in my 1 hour video. And guys, if you want to learn more, I'm sorry, more from me, then you are more than welcome to become part of my plus courses. I'm saying it again and again, my name is Dr. Pooja Sharma, with 8 years of experience as a scientist. Go find me on Unacademy. Go find me on Unacademy's platform. I teach you guys physics, chemistry, biology, 9th class and 10th class, 7:30 p.m., 8:45 p.m. every day. Okay, and guys, here is my referral code. Here is 10% discount that you will get. As I said, I take care of your physics, chemistry, biology classes. And go check my reviews, check my people, they follow me and what they say about me. And if you want to learn really well science, go ahead and subscribe on Unacademy. Okay guys, I will see you very soon in my plus courses. Please go ahead and sign up. And with this, end this session. Do you want to keep your connotations? All right, keep it. With this, I end this session. You guys have a wonderful time. Thank you so much.